Okay, so obviously we're going to be talking about the structure of DNA and RNA and the whole process of protein synthesis. As always, I skip the crap. So here's the idea. First, we're going to talk about the components. In other words, we're going to talk about what nucleotides are, what is the difference purine and pyrimidine bases. So this is bullshit. There are no purine. Those are bases, not purine bases. Okay? So let's talk about the bases first. There are two. Okay, before we talk about the bases, let's take a look at the nucleotide. Okay, check this out. So this is a nitrogenous base, the blue one. That's obviously a sugar, and this is the phosphate. Okay, so terminologically, this is a nucleoside, and this entire thing is a nucleotide. Within the same molecule, okay, sugars and phosphate are the same. So for instance, if it's DNA deoxyribose, RNA ribose. So effectively, the only thing that distinguishes one nucleotide from another is the base. Crap. Okay. Two types of bases, pyrimidine and purine. Okay? The reason why we call them nitrogenous bases is because of plenty of the nitrogens that are included in those cycles. For your information, not for the exam. The compounds that contain within a cycle an atom that is other than carbon, like these ones, we have nitrogens as the members of the cycle. They are called heterocyclic compounds. Just FYI, okay? So these are bases. The pyrimidine bases, there are a total of 3 that can be found in either DNA or RNA or both. Cytosine is found in both. Uracil is found only in RNA, and thymine is found only in DNA. Purine bases are adenine and guanine. They are found in both molecules. You can see that pyrimidine bases, basically pyrimidine molecule, the bunch of bells and whistles. Okay? And same is true for purine bases. Now sugars. So sugars are pentoses, and I want to sort of count some stuff here. Okay? So, let's count carbons. It's carbon 1, 2, 3, 4, and 5. Same is true for the bottom sugar. Now, here's the thing. If you look at the numbers, you can see that each carbon is numbered not just 1, 2, 3, 4, 5, but 1 prime, 2 prime. See that? 1 prime, 2, 3 prime, and so on. So why? Numbers without a prime are for bases. Does that make sense? So carbon and nitrogen atoms in the bases, they are numbered 1, 2, 3, 4. You see what I'm trying to say? And since we need to number carbons in the sugars, we use 1', 2', 3'. Okay? The biggest difference between these two molecules, and it's a very important difference, is this. Okay? So you see a hydroxyl group here? This hydroxyl group is responsible for relative instability of RNA molecules, but I think I should not let myself go into the weeds just yet and talk about why this is so. Okay? Now, since we learned about the bases and the sugars, we are ready to learn about a nucleoside, which is a sugar and the base when they form a bond. Okay? So nucleoside is when a nitrogen atom in a pyrimidine or purine, in other words, nitrogen atom in a nitrogenous base, okay, forms that N-glycosidic bond, and it bonds to the carbon 1' in a sugar. Okay? Make sense? So we're going to talk about the naming in a bit, but this is what we mean when we talk about this glycosidic bond. Hydrogen and OH groups leave, forming water, and the bond is established between the 1' carbon and the nitrogen in the base. Question so far? By the way, this is another example of the dehydration synthesis. Now, nucleoside becomes a nucleotide when the phosphate group forms an ester or a phosphate ester with a hydroxyl group found on the 5' carbon of whatever sugar we're talking about. So, strictly speaking, we consider nucleotides that exist in RNA and DNA are only the ones that have phosphate group bound to the 5' carbon, okay? Let me ask you sort of a conceptual question. Do you think it is chemically possible to form a phosphate ester just like the one you see here, but with other hydroxyl groups? Sure, why not? It's just another hydroxyl group. The point is, if phosphate would be sitting here, we would not consider it a true nucleotide because we gave this entity or any other a definition. A phosphate must be on the 5' carbon. Does that make sense? Okay? So, if we go back here, you see that the 5' carbon in a nucleoside is a hydroxyl group. This hydroxyl group interacts with a phosphate, forming an ester bond. Okay? All good? So this table essentially summarizes the bases, the sugars, nucleosides, nucleotides and nucleic acids that are found in the cell. So you must know that difference, that difference. Other than that, the structures are pretty much the same. Now, we mentioned before how we name bases, right? Cytosine, uracil, thymine, adenine, guanine. When we start naming nucleotides, we change the ending of the name of the base. We put osin for purines and idine for pyrimidines. Let me illustrate my point. So this is adenine, the base. So we remove INE and add OSINE, adenine to adenosine. Okay? Guanine to guanosine. This is applicable to purines. When we talk about pyrimidines, so we had cytosine, we change the osin part to cytidine. Okay? Uracil, uridine. Thymine, thymidine. All right? Now, we obviously need to make a statement which sugar we use. So, if we use deoxyribose, we put deoxy in front of the name. If we use ribose, we don't put anything. So if you hear the term cytidine, it means that it's cytosine base bound to ribose. If you hear the name deoxycytidine, it's the cytosine base bound to deoxyribose. And finally, when we name the entire nucleotide, we also add that it has a phosphate. Okay? So the full name will be, for this guy, for example, the sugar is deoxyribose, so deoxy. The base is thymine, so we change it to thymidine, okay? Deoxythymidine. And the phosphate is on the 5' carbon. Deoxythymidine, 5' monophosphate. In some, I know, but make it a structural approach. You see what I'm trying to say? So, for instance, this one, okay? Let's play with this. illustration a little bit. Ribose. Do we need to put anything in front of the name for the nucleoside if it's not deoxy? We don't. So you have adenosine, right? So what I'm going to do now, I'm going to add the phosphate group, okay? What? Sorry. Okay, now that's phosphate. Are you with me? Excellent. What are the numbers of carbons? 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. How many phosphates? Just 1. So adenosine, 5 prime, monophosphate. Can there be more phosphates? Sure. We'll talk about it. But so far, does that make sense to everybody? All right. So usually, we just use letters because it's much easier. Because if you think about the molecule of RNA or DNA, nucleotides are different only because the bases are different. The sugars are all the same, and phosphates are all the same. So this table gives you a summary of naming for nucleotides found in the DNA and nucleotides that are found in RNA, okay? So, a convenient way, especially if you're an old school hard rock fan, to remember nucleotides found in the DNA, ATGC, sounds almost like ACDC, okay? Those are DNA. So, guanine, both. Adenine, both. Thymine, no. Thymine DNA, uracil RNA, yes. Okay. So when you put nucleotides, when you connect nucleotides, you form a sequence, and just like in proteins, this order of nucleotides connected to each other is the primary structure, okay? So both DNA and RNA are polymers, or multiple nucleotides, and in case of each connection between two nucleotides, the 3' hydroxyl group, the hydroxyl group that's located on 3' carbon, it's really hard to comprehend without a picture. We'll get there. Go on.
We just did all of this stuff in microbiology with the replication.
Sure.
So like you just kind of
good.
Like
PTSD, I understand.
Yeah.
And that 3' hydroxyl group forms an ester bond with the phosphate on the 5' carbon of another nucleotide. Okay? So as we mentioned before, the sequence is the primary structure. And it's important to understand that when we start adding those nucleotides to each other, the chain becomes directional. Do you vaguely remember protein structure that we were talking about last week? Remember we mentioned N-terminus and C-terminus. So a protein has an order of amino acids. Same is true for DNA and RNA. They have a certain order. I usually compare it to conga. You know, like people dance conga. A person faces the back of another person. This is how it's supposed to be. So that's the same idea. Now, check this out. So this is the DNA nucleotides. this oxygen, okay, attacks this carbon, forming a bond, another ester bond, with a 3' sugar. Making sense? So, next nucleotide would be added over here. Does that make sense? Okay? And as many nucleotides, you can add and add them. But when you're going to stop, one end is going to have 5' phosphate. Another end is going to have 3' hydroxyl. This is called 3' and this is called 5' end. And that shows you the DNA molecule has a direction. Okay? So this is tetranucleotide. Okay? Just like with peptides, when you have two nucleotides, we call it dinucleotide, trinucleotide, tetranucleotide. At some point, we start calling them oligonucleotides. So you can see that this part of the molecule consists of phosphate groups and sugars that go one after another, interchangeably, alternating. That makes sense? Good? Okay. So this is a 5' end. This is a 3' end. Good question. Which sugar is that? Is that a ribose or deoxyribose? Ribose, because there is an OH group at the 2' carbon. If there would be just H, that would be deoxyribose. So far, we good? Okay. Now, before we move on, a very quick, very short note. I want you to notice that there's in ribose sugar, there's this hydroxyl group pretty close to that phosphate. Just notice. Just that's it. That's the end of it. We're going to get back to this later. So far, I hope you all can acknowledge that I do not lie to you. Okay. So, instead of writing all those formulas, write sequence and letters. So, traditionally, the 5' end is on the left, 3' end is on the right. I have a silly question for you. You see that tetranucleotide? Can I write it this way? I can. Everybody will think that I'm a psycho, but it's traditionally 5' is on the left, 3' on the right. Just like with proteins, N terminus is on the left, C terminus is on the right. So which one is at 5'? Yep. So, double helix. Come on, I will try to make it a bit more fun. Okay? So the guy named Edwin Chargaff, I think he was Brit, based on the last name and the first name, should be Brit. Edwin. So he determined that if you take a DNA and you break it down and isolate adenines, guanines, cytosines, and thymines, the proportion of A is equal to the proportion of T, always. and the proportion of G equals to the proportion of C. Okay? So if you have DNA, one of my favorite sort of mental exercises, let's say you have DNA and you figure that there are 20% of A. What is the percentage of T in that molecule? Yeah. What is the percentage of G and C?
40.
No.
Oh my God.
I know, 30, of course. Because 100% is total, you already got 20 and 20 out, you have 60 left, and you're gonna have 30% for G and C. So this, I think he got Nobel Prize later, I'm not entirely sure. But two douchebags, a decent guy and a woman, started to work on the determination of the structure of a double helix. By douchebags, I mean James Watson and Maurice Wilkins, that is not mentioned on that slide. Wilkins was a pretty misogynistic guy. Francis Crick was actually a pretty decent dude. And there was Rosalind Franklin. She did X-ray diffraction experiments that allowed these two and Wilkins to propose that DNA is structured as a double helix. So Franklin's story is actually, I think, is one of the most tragic stories in the history of science, because she was really a driving force behind the experimental part of the work. She crystallized DNA, did X-ray diffraction experiments, determined that it was basically, based on the results, she said it's got to be a spiral. And 5 years later, after the experiments, she died of leukemia. Because she was an X-ray person, she got exposed to X-ray so much that she got leukemia and died. And as you know, Nobel Prize is not awarded to dead people. What I think is the greatest injustice to her, the famous paper that was published in April of 1953, in which Watson, Crick, and Wilkins proposed the structure of DNA, contained only those three last names: Crick, Watson, and Wilkins. Franklin's name was not even on the paper with the results that she got. So their idea, all of them idea, was that DNA is organized as two strands forming a helix, where on the outside are sugar phosphate backbones, and on the inside are bases. The bases form pairs. A pairs with T, and G pairs with C. So here you see how it can explain the whole idea of equivalent ratio, right? Does that make sense? So adenine forms two hydrogen bonds with thymine, and guanine forms three hydrogen bonds with cytosine. Okay? Well, kind of a stupid question, which of these two pairs, in which of these two pairs the bases are bound to each other stronger? GC, yeah, because there are three bonds, right? So it actually has profound implications for biology, because if you have DNA molecule that has a lot of GC pairs, then two strands are going to be kind of, sticking to each other strongly. If you have a lot of ATs, not so much. You can separate them easier. Okay. Now, I want to add to this statement that adenine and thymine, only adenine and thymine form a pair, normal pairs. You can encounter a situation when in the DNA molecule, A is across from C. They're not going to form any substantial connection. They just against each other. It's not normal. That's what I say by, normal pairs. So far we figured that the strands in the DNA are held together by hydrogen bonds. There are two strands, and they wind around each other like this. Okay? So, this blue ribbon represents sugar phosphate backbone. Okay? Now you can see that this spiral, this helix, it is not symmetrical. I mean, it is, but it's not like staircase. It's a little bend. Do you see that this gap, for example, is bigger than this gap? So these are grooves. Major groove and minor groove. Just FYI, it's structural elements. That's where proteins bind. Okay? So, complementarity rule. Let's put letters next to the existing ones. A. Keep going. C.
A, G, G, T, T, A, G.
Now, 5 prime, 3 prime. Maybe I'm jumping ahead of a train, but what should I put in the bottom?
3 to 5.
What do we call it when one strand goes, okay, let's say this way, and another goes this way? Are they parallel? They are anti-parallel, which means think of them think of a road. Like you're driving on Route 8, yes. You drive south. There are several parallel lanes that go south, all of them. But if we look at the entire highway, lanes that go south are anti-parallel to the lanes that go north because the directions are opposite. They go next to each other, but the directions are opposite. So strands in the DNA are anti-parallel. Well, let's say a few words about replication. This actually gives me an opportunity to digress and give you the best analogy that I have ever invented. And I think it is better than any other analogy for genetic information. So, here's the story. Imagine, it's probably going to, if you heard, just don't spoil. Your DNA is a recipe book. Okay, mine's better. I guarantee you mine's better. Or they stole it. I'm just kidding. So it's a recipe book, not just a recipe book. It is the recipe book. It's a family recipe book. There are like 20,000 different recipes. Okay? Right? So thousands and thousands of pages written by 4 letters, A, T, G, and C. If you want to give this book to your kids when they move out of the house, you need to copy that book, okay? That would be replication. Okay? I'm going to do this. So 2 copies of the same DNA. Okay? All good? This is replication. Now, it has 20,000 recipes. You're probably not making all of them every second of your life or every day, right? You're making the recipes that you need. So imagine there are people, friends, coming over to your house. And it's a pretty motley crew. So, vegan, a Jew, an Indian, a lactose intolerant, and a regular person. Sounds, after that, I should probably say that at least walk into a bar. But they're not walking into a bar, they're walking into your house. So, you need to cook something that is vegetarian, that is without lactose, this, and that. Okay? Make sense so far? So this book is enormous. It's huge. You're cooking like 5 dishes. Are you going to hold the entire book into the kitchen because of 5 dishes? Probably not. What you're going to do to get the text of those recipes in the kitchen? In the real life, like you have a book, you don't want to bring it into the kitchen because you're afraid it's going to get spoiled. Huh? Sure. Scribble on a piece of paper, probably not, probably take a picture on your phone, right? Make sense? Okay. Good? Transcription. Now imagine you come into someone's house and they're like, oh, I have an exciting thing for you. What are we having for dinner? We have these pictures of the recipes. You're going to get pissed, right? So you are feeding your guests, not with a recipe, not with its picture, but with actual dish, a protein. That pretty lousy analogy was intended to illustrate that practically every function within the cell is carried out by a protein. There are some exceptions, but largely. Okay, that's translation. Sounds good? Why do we have 2 strands of DNA? Go on. No. No. If you take a DNA molecule, that's chromosomes. That's 2 chromosomes. But each chromosome is a DNA, and there are 2 strands of it. Let me kind of guide you through. If you take 1 strand, hypothetical 1 strand, can you read it? Yeah. Can you hypothetically extract the information? Yeah. Why do you need a 2nd strand? But why? Because just cool. Why not 3? Why do you have a 2nd of something? Yep, that's it. It's back up. It's back up. And why I say my analogy is great, when you're done with cooking, what are you going to do with those pictures of recipes?
They're gonna sit in my phone.
Okay, I'm an old guy, I've heard that there's an app in which like photos can disappear after a certain period of time.
Oh.
Whatever.
Yeah.
Sure, but my point is, you don't need them. If you want to free up the space on your phone, you can destroy them. You still have a recipe. Make sense? RNA is unstable. DNA is very stable. Okay? Because that's That's your book. Okay? Does that make sense? All right. Cool. So here's what happens. First, you need to separate the strands. Think of it as opening the book, okay? So strands are separated by an enzyme called helicase, okay? So you see the strands are separated, and then each strand, each parental strand is individually copied, producing another strand. So in this scheme of things, the purple strands are parental, the blue strands are daughter strands, and you can see that each new double-stranded molecule contains one parental and one daughter, one freshly synthesized strand. This is called semi-conservative replication. We good right now? Okay. So the enzyme that catalyzes that synthesis, the enzyme that adds the strands, is called DNA polymerase. And what this enzyme does, I'm going to go all the way here. No, even earlier. Yeah, this. It forms what we call phosphodiester. Why phosphodiester? This is a phosphoester. Does that make sense? This specific part is also phosphoester. So I have one phosphoester, another phosphoester. This is why we call the linkage between two adjacent sugars phosphodiester linkage or phosphodiester bond.
Why is there a double bond from oxygen to phosphorus?
Just structure. It is like it is how it's organized. A phosphate anion, it's just structurally there are two bonds. Okay? There's no like big chemical reason, just
the way it is.
Yes. So the complementary base pairing, this is the chemical view of a problem. It says that it ensures correct pairing, correct placement of strands. It is true, but you have to think, so my question is, are there entities on Earth? You may not know the answer to it. Are there replicating entities on Earth which informational molecule, which recipe book, is not double-stranded but a single-stranded DNA? Which are those entities? Viruses, exactly. Single-stranded DNA viruses. I'm not calling them organisms because they're not alive, but it's beyond the point. So you see, evolution selected for a double-stranded DNA as the best molecule to carry genetic information because there's a backup and it's very stable. To give an idea about stability, I can tell you, you can leave DNA in water, like when you isolated it, or in a buffer, on the table overnight at the room temperature, nothing's going to happen to it. you isolate RNA, and it starts breaking down right immediately. Like it starts breaking down. You do everything on ice, and as soon as you isolated it, you immediately freeze it because it is very unstable. Now, what is the difference? Let me go back. Not here, but all the way down here. Yeah, this. So this is RNA molecule, right? You see, remember I told you to remember about this OH group? And remember, we discussed that in DNA molecule, this oxygen wouldn't exist. So basically, the proximity of a 2' OH group to the phosphate results in instability of that connection, of a phosphodiester bond in RNA. Does that make sense? And now, this OH group is present in every sugar. So every diester linkage in RNA molecule is unstable, which means RNA molecule is ridiculously unstable. Okay? I wondered if you would ask me a question, what about RNA vaccines? No. Why are they stable? Modified RNA. They introduce slightly different sugars, slightly different bases, and that makes it much more stable than like the regular cellular RNA. Just do modifications. So, as we discussed before, the first step of gene expression is transcription, right? So, during transcription, DNA uses a template to make RNA molecules, okay? Now, there are 4 types of RNA. Well, okay, for our intents and purposes, 3 types that you should be aware of. The one that you just saw, this, is a transfer RNA. And 2 others are messenger and ribosomal. So those are 4 main differences between DNA and RNA, and they are absolutely legit. So ribose instead of deoxyribose, I'm talking about RNA. Uracil instead of thymine. Single-stranded in living organisms. Viruses are different. And RNA molecules are smaller than DNA molecules, which is, no shit. Because, as we discussed in my analogy before, you're not taking a copy of the entire book in a kitchen. Each RNA molecule is just a photograph of 1 page. So yes, they are smaller. So, messenger RNA. Can't emphasize it enough. It carries information. That's your pictures on the phone, okay? Transfer RNA. Its function is to match a codon, learn what it is, with the amino acid. If I would extend my analogy, transfer RNA is essentially labeled container with raw food. Okay? So you look at the container and it says meat. Okay, I know what's inside. I need meat for that recipe. Okay? Ribosomal RNA does the cooking. Now, this is where my analogy fails. Okay? Ribosomal RNA is the part of a factory that produces protein, in other words, ribosomes. So this table summarizes those different RNA types, with the ribosomal being most abundant, which is logical, since, ribosomes, there are a lot of them. And this is the structure of tRNA. So I mean, this is how it actually looks like, but it's a bit useless. So we're going to use this structure in our discussions. The structure is often called cloverleaf, yes, cloverleaf structure. And there are two notable parts: anticodon loop and the acceptor stem. Acceptor stem is where the amino acids bind. Okay, we're good so far? So, protein synthesis, I'm an old school person. For me, transcription is transcription, translation is actually protein synthesis. Because not every transcription will proceed. Not every RNA that is produced as the result of transcription will be used in translation. Okay? We'll probably get back to this. So this is the schematic representation. You've got DNA. How many strands? So double-stranded DNA. And then we get messenger RNA. How many strands? Single-stranded RNA. And then that mRNA is used as a template to produce a protein in the process of translation. We good so far? So there are two strands in the DNA. But during transcription, only one strand, called template, is used as a matrix for RNA synthesis. Okay? Only one strand is actually copied. You follow? Go on. Yeah.
What rRNA is.
What do you mean? Ribosomal.
Yeah.
You mean, okay, fair question. Let's add it here. I'll just explain. Well, in the ribosomes, it's weirdly shaped. It is a structural component, and actually, since you probably remember ribosomes, what they do, they connect to amino acids, okay? Serves as an enzyme forming peptide bond, okay? Peptidyltransferase. Now, you youngsters have no idea how explosive was the discovery that it is RNA that facilitates the formation of the peptide bond. It was insane. Because before that, I was taught in school that enzymes are proteins. Turns out there is a decent chunk of enzymes that are RNA molecules, not proteins. There were Nobel Prizes given for this. The guy who received one of them is still alive. He's in University of Colorado Boulder. Like he's a very famous dude, Thomas Chan. So,
Yeah, I thought enzymes were proteins.
there are some that are not. So let's get back to this. Again, I'm going to annoy the living shit out of you. How many strands are in DNA? Double-stranded, right? And mRNA? So this image explains it, right? You have template. One of the strands is considered to be template. It is used as a template to make RNA. Pretty straightforward, isn't it? Why do we need a second strand? Well, yeah, but in the process of transcription. Hypothetically, can I just make a single-stranded DNA molecule? First of all, I can. And mix it with RNA polymerase and have RNA. Nope. Turns out transcription as a process requires two strands. Only one is used, but you gotta have two. Okay? So this shows you how this enzyme, RNA polymerase, slides along the template strand. So that's template, okay? And synthesizes messenger RNA. Okay? Good? So, let's say we're going with this DNA molecule. What's going to be the sequence of the complementary RNA? Wait. This is the,
Oh, yeah.
Yes. This is the, yes. This is, I use this sketch in my tests. So yes, it's uracil. Okay? Where should I put numbers on the B? Yes. If there is a duplex, it's called a duplex. If there are two nucleic acids that are complementary to each other, whether it's two DNA strands or DNA RNA strand, they are always antiparallel. Structurally, it cannot happen any other way. Okay? Good? Let's move on. So, genetic code. So these triplets of nucleotides called codons. Something that I want you to process. Codons, I'm going to give you some hypotheses that people were exploring in like 1950s and 1960s. One of the first questions was how many nucleotides you need to encode one amino acid. That problem was solved very quickly. We'll get to why 3, okay? And second question was, do codons overlap? It was not a trivial question. So imagine this. Hear me out. Imagine you have this sequence. I'm just making it on the spot. Okay? U. Right? So the question was, is it this, the codons, or is it this, or they overlap in some other way? You can imagine that. It's not like a language. It is a language, but there are certain rules. So, long story short, they don't overlap. Okay? And how scientists sort of decided that it is 3 nucleotides that you need to encode for one amino acid. So we have 4 nucleotides, right? A, T, G, C in case of DNA, or A, U, G, C in case of RNA. If you hypothetically decide that one nucleotide encodes one amino acid, how many amino acids you would be able to encode? Dude, no. One letter for one amino acid. How many different letters do you have? 4. That's it. 4 amino acids. No way. What about 2? Okay.
2.
No. Okay. If you have 4 letters, how many 2-letter words you can make? 16.
Oh,
If you have,
took statistics so long ago.
oh, it's not statistics. Can I show you one of my favorite tricks? You're familiar, digression, but you're familiar with the logs with the code?
Yeah.
Yeah.
We did that in statistics.
It's combinatory, but okay. So if you have 3 numbers on the log, 3 dials, how many different combinations can you produce? Each dial has numbers from 0 to 9. What is the very first code on that log? 0, 0, 0, right? What is the very last code on that log? So 999 plus 1, 1,000. Does that make sense? You can have 1,000 different combinations. How many positions? How many dials? 3, right? We're talking about the log now. 3 dials, right? How many different numbers are on one dial? 10.
It's like 10 to the third power.
Yep. Okay, does that make sense? Good? How many dials we have in one codon? 3, right? 3 letters. How many different letters we can have in one position? 64. 4 in the power of 3, okay? When someone showed it to me, I am mathematically dumb. So it's like, I am a simple person. That fascinates me, okay? Good? So, we have 64 different nucleotides. We have 20 amino acids, okay? We have more 3-letter words than we have objects to name. Does that make sense? Even considering, hear me out, we need the words for stop codons. We need a word for the start codon. Even with this, there are more words than objects. And this is why the genetic code is called redundant. But my favorite synonym for this is degenerate. It is a term. Degenerate means that the same amino acid can be encoded by different codons. Look at my favorite leucine. 6 different codons for the same amino acid. Am I making sense? Good? So I compare it with synonyms. They are, and actually there's a term synonymous codons, which means that they encode the same amino acid. So this is how the code works. So like CCU encodes proline, AGC encodes serine. You, of course, for the exam, you must memorize the entire table with all the codons and all the amino acids. Dude, no. Like, forget it. It's going to be provided. Come on.
Or you just hate it.
Those things are not mutually exclusive, technically speaking. Anyway, so no, of course I could never understand the point in memorizing stuff. I remember exactly 2 codons. I remember the start codon and the codon for proline, which is CCC. That's it. If I need, I can Google it, okay? So, what is the sequence here? What's the first codon? What does the first codon encode? Huh? GCC, GUA, and GAC, aspartic acid. That's it. Okay? Does that make sense? Now, we kind of did not talk, and I want to briefly mention how this whole, well, we'll have a chance when we talk about translation. So we need to figure out how in translation you can insert the proper amino acid next to proper codon. Okay? Good? So effectively, we are looking at this. So this is mRNA. Okay? These molecules are tRNA. And this is anticodon. So far, do you follow me? So if the codon is AUA, then the complementary anticodon will be UAU. Okay, does that make sense? All tRNA that carry anticodon UAU carry an amino acid, isoleucine. If you would try to match this anticodon with this codon, you would fail. Does that make sense? So I want you to understand that this ribosome, it is not some tiny little, full of tiny little Oompa Loompas that just fish out specific tRNA molecules and just, check if they fit or not. It's all bumping. Like if tRNA molecule is there and it fits, it's going to be inserted. The amino acid, I mean. If it doesn't fit, it's going to be thrown away. It's going to dissociate. It's going to float away. Okay? So basically, the magic happens here. The peptide bond is formed between the growing polypeptide and an amino acid. Okay? And what's interesting, I love this part. Technically speaking, this entire polypeptide chain is transferred on that amino acid. So the growth is not happening at the end. You see what I'm trying to say? At the very free end. It happens within the ribosome. Okay. So this synthesis goes on and on and on until the ribosome encounters one of the stop codons. Stop codons are matched not with tRNA, but there are so-called release factors that bind to anticodon, not to anticodon, that release polypeptide. Okay? All good? Now my question, one of them, is what do you think the advantage of having several different codons for the same amino acid.
tRNAs carrying the anticodon can provide
Say it again?
the whatever they're carrying. Because you have the
Wait, hold on. Go back to our idea of a recipe. Okay? So it's a weird language that we're looking at. But think about this. Imagine that in your recipe, there is a typo, which is a mutation. Okay? Can it ruin the recipe? It can. Take a look at prolines. By having 4 different codons, if there is a mutation, and I acknowledge only if there is a mutation in the 3rd position, it's not going to affect which amino acid will be placed in the protein or which component will be added to the dish. Does that make sense? You see the idea? Yeah. So basically, it reduces the influence of mutations on the protein structure and therefore function. So protein polypeptide is released. When it is released, it gets in a cytoplasm usually and starts forming an energetically ideal shape, okay? Forming secondary structure, then forming tertiary structure, and potentially quaternary. Okay, does that make sense? But here's one interesting story about this. A lot of proteins, they cannot form a shape by themselves. Do you follow? There's a special kind of proteins in the cell called chaperones, which shape the proteins. They assist them in folding properly. What do you think happens to a protein that is not properly folded? Okay, so buy something from IKEA, spend like 5 hours trying to put this thing together, and it just looks ugly and dysfunctional, huh? Not only this, do you want that piece of furniture in the mid? You're gonna get rid of it. So misfolded proteins are usually destroyed, well, recycled, okay? Now this illustrates the whole concept of, the template strand, the copy of it in the form of mRNA, 4 individual tRNA anticodons, and the corresponding protein synthesis. So you should be ready to do this. Hear me out. You're given the DNA strand, you complement it with RNA strand. Can you do that? Or DNA, complementarity. Can you? Probably. You're given an RNA strand, you should decode it into amino acids, and I'm going to give you the table. Can you do that? I think so, right? So, why do we care? A lot of antibiotics, as you can see here, they inhibit protein synthesis. Now, bacteria and humans have very different structure, well, not very, but different structure of ribosomes, which means you can make a drug that will inhibit bacterial ribosome, but not human ribosome. So protein synthesis inhibitors are one of the best antibiotics that humans have. Now, recombinant DNA. Oh, I think we are isolating. Don't remember when we're isolating DNA, maybe next class. Anyway, so what is recombinant DNA? You take a DNA fragment from one cell and put it in the DNA in another cell, okay? So the most common way to work with recombinant DNA is to get the gene out of some other DNA and put it in so-called plasmid in bacteria E. coli, okay? So how does that work? So look at this. What is a plasmid? Plasmid is a small circular DNA found in bacterial cells, okay? You can treat it with a special enzyme called endonuclease, and you can cut it open, okay? So far? No, it's okay. So you open up the plasmid. Good? You mix that plasmid with the gene of interest, okay? And the enzyme called ligase connects the gene of interest with the plasmid itself. Now, I'm going to briefly highlight what sticky ends are. When the enzyme called endonuclease breaks down, let me just make up something. Okay? That's DNA, right? So sticky ends mean that the endonuclease will cut, for example, here and here. Does that make sense? So one end of the plasmid will look like this, and another end of the plasmid will look like this. These overhangs allow you to do pretty much the same procedure with the gene of interest, and the gene of interest, only the gene of interest will fit into those sticky ends. So now you have plasmids that contain that gene of interest. And you can force bacteria to make whatever protein is encoded in this gene. Okay? A whole bunch of proteins that are ridiculously expensive to isolate. And how are you going to isolate human growth hormone? You need humans, a lot of humans, which sounds very Nazi, so much easier to produce it independently. Okay? Now, there is one problem with production of human proteins in bacteria. We go in completely in the biology direction. What is the biggest difference between humans and human cell and bacterial cell? What do human cells have that bacterial don't? Huh?
A membrane-bound organelles, a nucleus.
There you go, a nucleus. So the whole process of protein synthesis in human cells is more complicated, and proteins are usually a bit modified with some bells and whistles. Bacteria can't do that. So I'm going to challenge you. It's not an easy question. Why do we use bacteria? Single-celled, grows super fast, easy to handle. Can you think about a eukaryotic organism that single-celled, grows very fast and fairly easy to handle? I'm going to give you a hint. You probably can find it in your kitchen. Huh? Yeast, yes. Once you do it in the yeast, the eukaryotes, they produce proper proteins, and as a matter of fact, most of that stuff is produced in yeast rather than in bacteria, okay? Now, polymerase chain reaction, PCR. I forgot what the name of the guy who invented it was. He, of course, got a Nobel Prize for it. He is very well known in biology community for being a total douchebag. Even polite biologists, oh, that guy, oh yeah. Kerry Mullis his name. Anyway, so here's the idea. You can try and replicate the process of replication in a test tube and do it for multiple rounds. So if you take a DNA molecule, split the chains by heating it up, breaking those hydrogen bonds, copy each of the strands, okay? Then split it again, copy each of the strands, then split them again, and copy each of the strands. So if you start with one molecule, how many DNA molecules do you have after the first cycle?
2,
Second?
4,
Third?
8,
How many molecules are you going to have after 20 cycles? 1,048,536. When I was a kid, I was bored and did math when I was bored. So basically, after 40 cycles, you have more than 1 trillion copies of the original DNA. Does that make sense? And that many copies of the original DNA can serve as a signal. You can detect it. Okay? Sounds fair? This is why we use PCR for various tests. For example, for the disease. Remember PCR testing for COVID? Extremely sensitive. Okay. So we can screen using PCR for very specific genes in a human being. Okay? For instance, we can screen for BRCA1, BRCA2, which are genes associated with increased risk of breast and ovarian cancer. We can check for other genes. So basically, you're familiar with 23andMe and Ancestry.com. They don't sequence the entire genome. There are certain parts of it associated with disease and such. So they basically run very short PCRs, very small PCRs on small number of markers within the DNA, and then tell you, oh, you 50% Irish or whatever, okay? Fingerprinting. So I mentioned before the term restriction enzyme. Remember? Let me go back to this. Here, restriction enzyme. Restriction enzymes cut DNA. That's it, just cut. And each enzyme has a very unique, specific sequence that it will cut, and nothing else. Knowing those sequences and having more than one restriction enzyme, you can effectively cut the DNA and make sort of a map of the fragments. The irony of that particular slide is that it illustrates the point that chemists should not be let anywhere close to biology concepts. Because the dude here, it is not a restriction. This is not DNA fingerprinting. This is sequencing. It's a completely different process. It's classic Sanger sequencing. It is not DNA fingerprinting. Okay, does that make sense? So, speaking of sequencing, human DNA is sequenced. So think of it of this as a recipe book written in the alien language, which humans decoded. Interesting thing is, if you will try to look up how many genes are in the human genome, the answer will be There are like so many known genes. Not all genes are known. Not all genes are attributed yet. Okay? So, 21,000 known genes. Okay? So 21,000 genes comprise only 3% of the total DNA. So what does that mean in terms of your recipe book? It means that only 3% of your recipe book contains recipes. Everything else is a bunch of gibberish. Sometimes this gibberish is in the middle of your recipe, so you have to sort it out. Some of it is the term that we used before was junk DNA. This term is not used anymore because a lot of those parts, they may not be transcribed and translated as genes. They may produce regulatory RNAs, or they may be regulatory by themselves, and enhancers, and various different types of proteins. Okay? So this is a great question, because they're I know which answer they want. What do you think is the right answer according to the authors of the book? Yeah, that. But yeasts are also used for recombinant technology. Does that make sense? Now, what are we going to do? We're going to take a break.